Hello, everybody. Welcome to our 11th Bio Art Mixer. I can't believe that we have done already 11 of those, but I'm very excited to have it now and to welcome our guests who I'm going to introduce just in a second. But before that, I just want to say a few words about the Bio Art Mixer in general and explain what it is and how it started, but really short for those who have never been here. And I see that there are some people the new faces. I see new faces. First of all, I'm Boriana Rosa. I'm associate professor in transmedia, and we have started this BioArt Mixer, I think 2 years ago with Dr. Heidi Henley, who is a professor in biology department at SU. And the idea was to create a community that is informed and profoundly interested in the field of bioart, and not only bioart, but also the crossover of science and art. I am an artist, and some of my projects are within this field, so I have professional interest and experience in it. Professor Henley, she has been interested in art forever, I think. I think this kind of idea was very organically coming from our practice and from our interests. Our future plans is to create a bioart show in which we will show mostly art and research created within New York State and a little bit above, but mostly within the United States. So Heidi, do you want to add something?
I think you're doing a really good job. And I have Bart here, so he probably wants to add something.
Okay, good. So again, I want to introduce our guests today. We have the opportunity to, like with these online meetings, it is easier to invite people who don't live closely. But at the same time, unfortunately, we are keeping the in-person contact in its full. But nevertheless, we're happy to have people who are not really, how is this called, geographically close to Syracuse. And first I want to introduce Professor Jennifer Willett. She is an associate professor in the University of Windsor. And she is, besides being an artist, she is a Canada Research Chair in Art, Science, and Ecology. And as I said, associate professor in the School of Creative Arts at the University of Windsor. She has been internationally successful as an artist and researcher, and I know a lot of her amazing works that have been exhibited and researched and made outside of Canada within the field of bioart. She had worked with Sean Bailey as a collective which was called Biotechnica, and she completed her PhD in the interdisciplinary humanities program at Concordia University. She had been also working as a researcher in the Genomics Center in the University of Leiden. And she has opened in 2009 the first biological art lab in Canada, which is called Incubator, hybrid laboratory of the intersection of art, science, and ecology. And this is at the University of Windsor. And I assume that some of this work will be also part of her presentation. So she will talk about her teaching ideas and the way how this incubator lab works.
Thank you for having me.
Thank you for coming. Richard Pell is our second artist and professor who participates in the BioArt Mixer today. He works at the intersection of science, engineering, and culture, and has worked in a variety of electronic media from documentary video to robotics to bioart, and also museum exhibitions. He is the founder and the director of the Center of Post-Natural History, which is an organization dedicated to the collection and exposition of life forms that have been intentionally altered. And this, I assume, again, will be a big part of what he's going to be presenting today. So the Center of Post-Natural History is a permanent museum in Pittsburgh, Pennsylvania, and it is also a traveling exhibition that have been around the world in many different places, including a major exhibition at the Wellcome Collection in London. Also, Richard Pell is an associate professor at the Carnegie Mellon University, and he has been doing a lot of other artistic work that, again, has been presented around the world. Successfully. Now maybe Heidi will introduce our next guest.
So our scientist for the evening is Dr. Joseph Paulsen. We're really thankful and lucky to have him. He's in the Bio-Inspired Institute and also the physics department here at Syracuse University. He did his undergraduate work at St. Olaf and got degrees in both math and physics, and went on to do his graduate work and PhD work at University of Chicago in physics. And now we're lucky to have him here at Syracuse University, where he has been awarded, I think pretty recently, an NSF Career Award, which is a really big deal in the sciences. And he does some really awesome work on wrinkling, and I think he's going to talk about today. And one of the reasons I got really into it is because his beautiful images of these different substrates and how they can wrinkle under different conditions. And I think he's going to give us more specifics on that I'm really excited to hear.
Thanks for having me.
I also wanted to introduce Professor Edward Morris, who is going to be our moderator of the discussion, which will follow the presentations. First, we'll start with Jennifer, then we'll have Joseph, and then we'll have a break. We'll have about 10 minutes break in the middle. And then we'll have Rich Pell and a discussion, which will be moderated by Professor Morris, but you are also welcome to put down your questions in the chat meanwhile. So these questions can be integrated in the discussion. And again, if you have new questions during the discussion, the chat is one of the tools that can be used for that. Ed, do you want to add something to the protocol?
Yeah, I just want to emphasize the chat. So, you know, the BioArt Mixer, one of the impetuses for the BioArt Mixer was to actually mix, physically mix and follow the talks by having drinks and talking together. We can't do that because of COVID. Welcome back to Zoom, everybody. But like the way we can kind of approximate that is the chat. And in the past, when we've moved on to Zoom, we've had some pretty amazing, robust discussions in the chat. And so I just encourage you to use that. I'll make some prompts in there and just follow along in the chat and add your thoughts there. It's a great way to have the conversation as we go.
Okay, so maybe we can start with the first presentation by Jennifer Willett.
Sure, thank you for having me. Let me just share my screen. So the first thing I would just really like to do is to say thank you to Boriana for having me here today. We've known each other a long time and we sort of come up together through the BioArt community. And so it's just always a pleasure to see you again. And I really value the community that you've built with these mixers. And I'm really glad to be here to participate. So I'm Jennifer Willett, and I am the director of a lab called Incubator Art Lab. And today I'm going to focus my talk on talking about the lab and some of the activities that we do, but I will also show you some of the work that I do, as well as I'm going to give you a little peek of what I've been doing since the pandemic hit. So I'm at the University of Windsor, and we're in the city of Windsor. This is the view from the end of my street, and we're looking across the Detroit River at Detroit, downtown Detroit. So it's important that you sort of understand the location that we're working in. We do have a site-specific laboratory that engages in our cultural and our ecological environment. So we're right on the border with the US. We are on traditional indigenous territories. We're a huge site. This is the #1 site of economic exchange across the Canada-US border. So it's a really like loaded political environment, as well as it's at the heart of the Great Lakes Basin. So it's a really like rich and ecology that we're working in. Windsor was an auto manufacturing town. I have to say a lot of that has really depleted over the years. So it's a little bit of a post-industrial city, but certainly reimagining itself in a variety of ways. But one of the things I really like about that history and my relationship to the town and to the university is that my students are really makers. They have real hands-on knowledge. So they've all taken a car apart on their front lawn. And so they're a little fearless about some of the things that I'm asking them to do in the BioArt lab. And I wonder sometimes if this history of tinkering and of like technical manual knowledge is one of the things that has allowed me to be so successful in this environment. I'm in the School of Creative Arts, and we have a new downtown campus that is part of a renovation and a revitalization project in downtown Windsor. So they turned the old Armories building into an arts complex, as well as the building next, they built a standalone building next to it. And in the front lobby of the new building, we have the BioArt lab. And this is a BSL Level 2 BioArt laboratory. And it has a floor to ceiling glass wall that separates the lab from the main atrium in the building. And we use this in a variety, we chose this space for a variety of reasons. It means we have a much smaller lab, which is fine because that's a lot less work to manage. But what it means is that we have a lot of visibility. And so I'm very interested in transparency and audience engagement in this space and making visible things that are often kept hidden. So casual passersby can see into our lab, but we're building the lab into a multimedia theater, which I'll show you a little bit later, so that we can do things like BioArt opera or BioArt performance art events that then live audiences could see biotechnological protocols and performances that can only happen in a BSL Level 2 environment. You can see the aesthetics are a little bit different. I'm interested in exploring what a laboratory can look like and who belongs there. And so it's really looks a little more like a domestic space. We've begun transforming this space a little bit further. We're putting a medallion on the ceiling. We're hiring a plaster worker to come and do some plaster work for us. So this was some of the concept sketches that I did when we were applying for funding. So we've just received funding to put in a multimedia theatrical components into the space. So it will have theatrical lighting and sound, video projection components, as well as video recording components. So the goal is that we'll be able to do these sort of strange multimedia hybrid events and then put them online as well as perform them for live audiences. In addition to that, we've been really fortunate to receive a second space. And this space is affiliated with my Canada Research Chair. And it is a downtown storefront studio space. And the goal of this space is twofold. One, the front area, which you can see at the bottom left here, is like a community engagement space or hub where we can host BioArt workshops for regular people. One of the problems with the lab is it's beautiful, but since it's a BSL Level 2 lab, you need 3 days of training to be able to go into there. So we wanted a space where we could just have school children, people under the age of 16, regular people come without that type of training. And the back area is more of a woodshop building area. I do a lot of built sculptural projects as well. And so we needed a space where we could assemble those projects. One of the things that's gonna be very interesting about this lab is in the front area, I have a little greenhouse that fits over top of essentially like a coffee bar. And that's gonna be the certified laboratory environment. I'm very interested in how artists and bioartists in particular can skirt the sort of boundaries of institutional bureaucracies to create new spaces and new avenues. So one of the things that I was thinking about for workshops is I wanted to be able to have workshops in the space, but I also wanted to be able to have coffee with my team or work on an art project that didn't have all the health and safety implications. So what we're doing is we're enclosing the lab in a greenhouse, and when the doors are open, it'll certify the whole room as a lab. And when the doors are closed and locked, it'll no longer be a lab. So this is the space. We just got it at the beginning, like around the pandemic, we were negotiating spaces. We were working with an architect. So I really just got the keys a few weeks ago, and we're just starting to move in now. The heat hasn't been turned on yet, and they're worried the pipes are gonna freeze. So it's been like, it's a really strange experience right now. You know, we're all sort of trapped at home. I have this gorgeous studio that I'm not allowed to work in. And to also have these really beautiful, positive, like, 10-year goals come to fruition at the same time as we're all sort of like depressed and at home and really concerned about the pandemic. So it's beautiful, but it's a little bittersweet. So this is my team. So what I've done over the years, and I was talking to Rich about us presenting in the same environment, I've created this bizarro institution around my practice that also sort of supports my practice. The institution itself has become part of my practice. I've been thinking a lot about like, what is a lab? How can you manage a lab? How can research groups produce research? What type of research do they have to produce? And so we've been producing artistic or research creation outcomes using a more traditional laboratory research group format. So we meet once a week, and we have different projects that we're working on. The projects range from my projects to events that we're hosting, to educational stuff linked to the class or to community engagement workshops. And then all these people come from all different backgrounds. I really hire for enthusiasm. I hire for imagination. I have to say I pay everyone. I do not accept volunteers in my lab. And we're trying to think about like community building and ways of working together that are maybe more collaborative. And so these are the people who are working with me this year. I have artists, I have scientists, I have philosophers, I have people from all different cultural backgrounds working in the same environment. And it's this like cacophony of voices and energy that has been very, very productive over the last few years. These are our students. We haven't seen them in person in a while, but again, you can see that there's like this type of vitality and energy that I try to engage with the students. And I really have folded my research and my teaching into this sort of enmeshed practice. So the students end up contributing to research and learning about how research can be produced. And then I end up sharing with them about what we're doing in the lab. I hire a lot of them, and then they also learn a lot about bioart through events like this one. So the lab is based on the premise of a sort of a fundamental idea that biotechnology might be or could be or is a technology of love. And that there's these complex interrelationships between different organisms, human and non-human organisms, within the biotechnological workspace. And that the type of love that we might be engaging with is not the romantic type of love that we might think of from a romance novel, but rather sort of a sense of like co-participating in the unfurling of the universe, a sense of mutual respect, a sense of collaboration and a mutually beneficial, but also mutually detrimental relationships. Understanding that love also acknowledges suffering and pain and death, and that those are not necessarily things that could ever be avoided, but something that has to be understood or acknowledged. So the goal of my research chair is to devise methods, artworks, texts, and technologies that allow audiences, species, and communities to collaboratively engage in imagining possible biotech futures. And these futures, I'm trying to avoid the sort of Hollywood dystopic movie. I'm trying to avoid the things that we see in the news media and sort of adversarialness. And I'm trying to imagine biotech futures that integrate feminist or post-colonial perspectives, futures that integrate advanced digital and biotechnologies, but also that look to historical knowledge and agricultural knowledge and indigenous knowledge. You know, so often we think of the future as like moving away from all of that, but actually I think that those things have to be reincorporated. Those knowledges are being lost. Community engagement, sustainability, love, joy, gratitude, and the acknowledgement of suffering. So to that end, we do a whole variety of projects. And we really, again, it's really about engaging and learning about new communities and sort of thinking about alternative ways of thinking of things. So we do all sorts of workshops and events. This is one we did a long time ago in 2010, where we took some of the Windsor students up to Montreal and we did a workshop with Tanny Duff and Stellark there. Sometimes the students in Windsor have sort of a downtrodden view of Windsor or the work we're doing there. And so I love to take them to big cities and allow them to engage with other artists. And it really helps them to understand that what we're doing is really exciting and that they're really skilled and they have a lot to offer. We do I curate a lot of shows and various different venues, that type of thing. I felt like if I was going to start teaching bioart at the university level, I also needed to build audiences and maybe institutional frameworks for bioart to be exhibited more in Canada. Because if you're going to teach people these skills, you need them to also have opportunities once they graduate. We do a lot of community engagement events. This is the opening of our lab, the BSL Level 2 lab. We had about 500 or 600 people come, and we did this like really bizarre cabaret kind of night called Feasting the Lab, where we did all of the things in the lab that you're not allowed to do in a lab until the night before, and then we got the certification the next morning. So we did shots in the lab, we danced barefoot in the lab, we did all those types of things, and then we had a big raucous party, and then the next day the paperwork was signed, and we can never do that again. We do a lot of projects that engage with our local ecology. So this is a project that's been postponed because of COVID-19. It's called the Invasive Species Fishing Trip. We're going fishing with researchers, but also with Indigenous groups, as well as with subsistence fishers to learn more about our local ecology. And we're gonna gain knowledge about notions of invasiveness. We're gonna look at that from an ecological standpoint, but also from a social and political standpoint, sort of parallels and fears of the other that we have for human migration, aligned with some of our fears around biological migration. So a lot of my work is about building relationships and integrating myself with different types of groups. Right now, we're really connecting a lot with the Healthy Headwaters Lab, which is a lab that really works very, very closely with farmers and Indigenous groups in our region towards making like real solution-based strategies for improving the water quality in our area. And we also work a lot with IOTA Institute, which is an art and technology creative agency that is very interested in furthering bio-art relationships in Canada. And then I'll show you a little bit of my work. So that's sort of an example about the lab. So within that framework, I also make a lot of art. And like Boriana said, I have been doing this a long time. So there's a whole bunch of series. So I just picked one of the most recent ones to show you. This is called Baroque Biology. And it's intended as sort of a science fiction that integrates biotechnology with sort of other metaphors that we don't usually link to biotechnology. So usually we think of labs as like sterile and clean and perfect and digital. And so I wanted to build the opposite. We built these like ridiculous Baroque Rococo objects that were feminine and playful, and that could also be like in a children's museum, and allowed them to become the sites for biotechnology. You'll also see that the species hierarchy usually seen in a lab is upset in this work. So I'll show you some of the digital prints. So the sculptures are in the front. I won't talk about this today, but in the back, I'll show you the digital prints. And this is a way to also learn better about how the lab works. So this is the very first maquette that we made. One day tinkering in the lab, there's some temporary tattoos and I put them on tops of book pages. And I was thinking about creating this little like vignette, these little paper theater shows where non-human organisms teach humans about their biological processes. So maybe biotechnology is about us listening and learning to the other microorganisms around us. So my team and I, we worked on this for a couple of years, and it started off really like lowbrow, and we gained a little more experience, and we created some recipes, and we practiced with different collage materials. We found that the amino labs, I enjoy working with existing technologies. I don't always have to reinvent the wheel. So this is a kit for like kids who want to learn more about how to grow GMO bacteria, and they have like a rainbow kit. So we bought the kit and we offered to buy a bunch of the medium, but they wouldn't sell it to us. So we reverse engineered the recipe for the medium and we created a rainbow of colors. We learned how to cast agar. And then we started creating these sort of like biological vignettes, again, sort of upsetting the hierarchy between humans and non-humans in the lab. And the final stage is I purchased these paper theater sets online. They're historical paper theater sets from hundreds of years ago, to then create like a stage for these organisms and their relationships to interact with. You'll see all the research assistants. So the group at the time, they all contribute, but they also get credit for the work they do on these projects. There's about 20 of those. So the last thing I want to do is just tell you a little bit about what I've been doing since the pandemic hit. So the first little while I was home with little kids. It was really intense trying to teach and be with little kids. And I got super down and I couldn't really do a lot. And my partner was like, Jennifer, it's a pandemic. This is BioArt's time. You got to get out there and make some work. And I'm really glad he said that because it really helped. And it helped because he's absolutely right. This is our topic. We have a lot to offer and engage with, but also for me to sort of deploy my usual strategies that I have for making art and for connecting with other people really helped me through that time. So this is a commission called COVID Suit. And I think I might rename it COVID Suit Mom, because she starts doing some mom things later on. And I made this suit that's decorated with the molecular patterns as well as the viral patterns of the COVID-19 on the suit. And then I put all sorts of reflective panels on it. And I did all sorts of performances and ran all over town, especially at night, because the light reflected off it was really beautiful. And then so we're now working on a video. This is a photographic series, and now we're working on a video that will sort of express this internal narrative of the strange COVID suit mom. And then so now she's started doing workshops and performances. So this was for the Bio Summit at MIT. And they offered me to come and do this gratitude offering for the organisms. And so the COVID suit mom talked her way into the lab at the university. And I hadn't been in there in months, and it was like dead in there. And I wrote this like eulogy to all the organisms that we had killed back in March, and talked about how we had to sort of learn to transform our relationship maybe with viruses as well, towards maybe some of these other more interspecies or entangled or love-based relationships. And it was really fun and really scary being in the building all by myself. And then the COVID suit mom started doing workshops. So this is when we decided that we missed our lab so much. We built a puppet paper theater maquette of our lab. And now on Science Gallery Detroit, it actually ends tonight, but we'll put it live on our website tomorrow. You can download this workshop where the COVID suit mom comes and she shows you how to build the lab. And then she shows you how to like pour weird things and do experiments in your lab. And people have been sending us photos and videos of those results. That's it. That's everything I have for you today.
Thank you, Jennifer. This was a wonderful presentation. It's extremely interesting, and I've seen so many things that you've done that I didn't know about. It's great. Now I think it's time for Joseph's presentation.
All right, I'm going to share my screen. So you should see my title slide coming up right about now. Right, everyone can see and hear me?
Yes.
Great, so first I want to, of course, thank the organizers for the opportunity to speak to this audience. It was really fun to prepare this talk sort of outside of my usual mode of presenting this work. Now, this is a talk about wrinkle patterns, and you see an example of a pattern on a very thin film on my title slide. So I'm going to get to my particular research in a little bit, but first I want to just say a few words about patterns in general. So as you all know, patterns are objects or designs that are made out of repeating a single unit over and over and over in a given interval. And so, you know, humans are very good at recognizing patterns and creating them. And so if I ask you what's cut out of this image, you can probably tell me, well, it's just one of these squiggly things, right? There's squiggly thing, squiggly thing, squiggly thing. And this example is a Greek meander, which happens to be on a stove in Bucharest. Okay, and so on the architectural scale, patterns are quite common as well. So once again, we see pillar, 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 pillar. I'm sure you can guess what comes next. Well, there probably were pillars there before, but I guess they're gone now. In any case, on my scale that I work with in the lab, we generate patterns in a much different way. But the same exact idea is there. You see all of these vertical stripes, and so you can tell me exactly what should be in this box that I've cut out. It's just more of the same, more of these vertical stripes. Okay, so what's interesting about these things for a physicist? So the really remarkable thing about these wrinkle patterns is that I didn't have to sculpt this thing out of stone. I didn't have to have it 3D printed. Actually, all the ingredients were very formless objects. I took what's essentially a very thin contact lens, just a portion of a spherical cap. I placed this on a flat bath of water, perfectly planar, and out pops this thing. Okay, and so nature has actually made this pattern for us. And that's a really powerful tool scientifically. In particular, nature decided how big each of these wrinkles would be. And the powerful thing about that is that just by looking at this wrinkle pattern, we can learn a lot about the material that we're looking at. So if the material is stiff or if the material is thick, the wrinkles will be large. If the material is thin or if the material is soft, the wrinkles will be very small. And so simply by taking an image, taking a ruler and measuring the width of one of my wrinkles, I can tell you, I can measure quantitatively a material property of this thing. And I'm a physicist, and so I like really, really general principles that I can deploy at all kinds of different scales and into all kinds of different systems. And so here's the big impact of this simple idea, is that I can take an apple, which is rotting on my counter, and I can take my ruler and I can measure those wrinkles, and I can tell you something about how stiff the skin is on that apple. I can take my fingers and I can squish my wrist. I can again take my ruler and measure the wavelength, the width of those wrinkles, and I can tell you something about how soft my skin is or about how thick my layer of skin is. I can take a single cell and I can lay it down on a substrate, and I can look at how that substrate wrinkles as the cell pulls itself across that substrate. And by looking at that wrinkle pattern, again, take my ruler out to measure the wavelength of wrinkles, measure the extent that they travel out radially, and I can tell you something about the forces that that cell can exert out of its own molecular motors. For the biologists in the room, I can take a vesicle and I can put it in a simple shear flow, and I can look at the wrinkle patterns on the surface of that vesicle. And again, I take out my ruler, I measure the width of each of these wrinkles, and now I know something about the bending rigidity of this lipid bilayer. So this is a really general principle that can be deployed at all manner of different scale to give really kind of shocking insight into the mechanics of any material. Okay, so that's really what I get excited about as a physicist, are simple ideas that work in lots of different settings. So let me tell you a few words about what we actually do in my lab. So a lot of these experiments, these get me excited about doing science, but I don't actually do biology. I don't work with vesicles. I guess I do leave apples, or my daughter leaves apples on the counter to rot. The experiments that I actually do in the lab are what we call model experiments. So I set up very well-controlled, very simple settings, and I try to extract simple principles from them that can be deployed across scales. And so what we do is we take microscope slides, we put them in a machine called a spin coater. The spin coater is very good at laying down a very thin, very uniform layer of, in this case, polystyrene. So the same stuff that packing peanuts are made out of. And then what we do is we take this coated microscope slide and we just very slowly dip it in a bath of water. And as we dip this slide into the water, surface tension peels the layer of plastic off the surface of the slide, and off floats an extremely thin plastic film. Okay, and so here's a photo of an undergraduate in my lab. This is Graham, and Graham is patiently dipping his substrate into a dish of water and waiting for that film to float off. Sometimes you can do this in just seconds. Sometimes it takes 10 minutes of very patient, very still hands to get this to work. And this is a photo of Anna, who is a summer intern, and she's taking an image of one of these floating polymer films. And all of the photos that I'm going to show you from my lab are all taken with consumer grade digital SLR cameras with macro lenses. So we don't actually have a single microscope in the lab, but we have these macroscopes, as I call them, which are cameras set up on various stages with home-built lighting to get the images that we need. Okay, so the next steps, I work with a lot of different geometries. And so the next step would entail putting the film in some different geometry to see how it reacts. So in this image that I'm going to show you next, we're putting the droplet on a curved substrate. So this is laying an initially flat, initially planar circular disc of polystyrene and laying it on a curved substrate. So we're just plopping it on like a little polar cap on top a spherical water droplet. And for a sense of scale, these are kind of big sheets. They're millimeters to centimeters in diameter, but they're extremely, extremely, extremely thin. These are on the order of 100 nanometers thick. So this is 1/1000th of the thickness of a hair on your head. Okay, so extremely thin, very floppy, and that's what puts them in this very wrinkly limit. So then if we remove some of the water from the droplet and force the sheet to be more highly curved around the object, we get even more kind of interesting patterns that come that we can look at and study and try to discern principles from. And in the remainder of my time, sort of in the second half of this talk, I'm going to tell you about one particular pattern that had myself, my group, and several other groups very puzzled for about the last 10 years. And we sort of finally made a breakthrough at getting at the origin of this pattern. And it's apparent in this image, but I'll be showing you clear images of it later on. Basically, these are sort of sharper wrinkles that come to a sharp point and then end. And the term that we made for these is crumples. So we're moving from wrinkles to crumples now. Okay, so here's kind of the motivation and the early experiments that got us along these lines. These are experiments where, once again, a flat, very thin, very floppy plastic film is laid on the surface of a water droplet. And here, the one variable is that we're changing how curved the water droplet is. So by pushing and pulling water out of this tube, we can change how curved that little meniscus of water is. So in the left image, you see what happens when the sheet is on a flat liquid surface. You just get a flat sheet without any features in it. In the right picture, when we curve this water interface, this starts to form wrinkles at the edge of the film. And actually, these are quite familiar to all of you if you've ever had a disposable paper plate. This is exactly what those wrinkles are at the edge of the plate. They're giving a little bit of curvature to the object. So these are tucking away extra material to allow the film to curve around the spherical form of the droplet. If you curve the droplet just a little bit more, you get something completely new. So now we don't have these regular wrinkles. We don't only have these regular wrinkles. We also have these on a longer period, these sharper, more focused features. You see about 10 or 11 of them throughout going around this clock face. And this was a complete surprise to the lab that found these. This was discovered at the University of Massachusetts Amherst, where I was doing my postdoc. And these just captivated me. To a physicist, the exciting things about these is symmetry breaking. You start with a very symmetric object that has rotational symmetry, and you break this symmetry by dividing it up in these kind of 10 regions. To an engineer, these are really important because it means that there's stress at these points. This is where the material is undergoing the most violent stretching and bending deformations, or at these sharp points. And if you're just plain curious, to me, this is just a beautiful response that has very little intervention. Again, it's just a perfectly flat, perfectly circular disc dropped on a perfectly spherical droplet, and this just pops out. It's just totally stunning to me. And wine. So these are wine storage tanks that were subjected to an earthquake. And so normally these are just big cylindrical barrels, but due to this earthquake and the seismic waves traveling around the storage facility, these tanks buckled. These are on the verge of failing. They buckled and they made these patterns on their surface. And if you just look and compare these to the wrinkle patterns that I'm studying in my lab, you can see, I think, quite a striking similarity. So this is really a kind of a robust response that happens across scales from centimeters to tens of meters. And again, just to stress, a general understanding of what's going on here, it was totally lacking at the time that I was working on these. So this is the big mystery. What are these objects and why are they there? And what do they tell us about the material or the geometry or about the physics? And so once I started seeing these objects, I started to notice them kind of everywhere. So I was doing a completely different experiment. This was an experiment where we were poking a film. So you take, again, you float a flat, circular, very thin disc on a bath of water, and then we're taking and just draping it over a little steel ball bearing. So we're lifting this thing up like a little tent. And as I play the video forward for you, you're gonna see these wrinkles forming. And once again, there's these little characters again, these sharp periodic features that are collecting more material than their neighbors, getting kind of deeper and deeper. And so kind of out of nowhere pops the same wrinkling to crumpling transition that I had been puzzled about before. So we wanted to see, okay, can we isolate these? Can we distill these down to the simplest possible geometry that we can think of? And to me, what I imagined is that probably the simplest geometry that could make these things is just a cylinder. So let's take a rectangular sheet, let's place it on a cylinder of water. Making a cylinder of water is actually pretty simple. You just take a little channel and you flood it ever so slightly with water and you get a little bulged up cylinder out of that. And then let's sort of compress this thing between two walls. And that's what's going to force it to buckle or to wrinkle and make these undulations. Okay, and so this is my postdoc, Yusra Timune, who was doing these experiments. And here's what she saw. So first, what I'm going to show you here is we're starting with a flat bath of water with a flat rectangular sheet of polystyrene on top of it. And first she's crushing it between these walls to form these wrinkles. And now she's curving the water into a cylinder. So she's slowly flooding this tank to go from a plane of water to a cylinder of water. Okay, and again, we see these things just kind of popping out from these smooth, kind of area filling wrinkles to these sharper, more punctuated crumples. So, okay, so these things just started showing up everywhere at this point. I was, playing with my daughter in the backyard in the summer, and I was noticing these things in a kiddie pool. So this is the inflated wall of a little swimming pool. And what I want you to kind of cue in on are these sort of repeated sharp elements that are kind of lining up along diagonals. And then, on another weekend, we were over in Geneva, New York, staying in a hotel on one of the Finger Lakes. And there was this sort of sunshade. And we were walking around at night with my family. And, there these things are again. There's this canvas that's draped over these support columns. And again, there's this exact same kind of repeated structure. You have this kind of diamond-shaped element, and it keeps repeating and repeating kind of along a diagonal. Okay, it's the same kind of microstructure that I was seeing in the lab. And you get your box from Amazon, which is like where everything comes from these days to our house during the coronavirus. And you get these little pillows that are used for packing material. And you can actually do a nice experiment with one of these things. If you grip it kind of loosely, you can get these this crumpling motif near the boundary of these little bags of air. And then if you start to clench your hand down to really push on the air inside of the package, they will transition into these smoother wrinkles that you're seeing in the left picture. So you can actually transition between these sharp wrinkles and these smooth crumples just by squeezing one of these air pillows that comes in one of these packages. So these things are really just kind of staring me in the face at every turn. And to kind of wrap up the talk, the big success story is that we figured out how to make them in the lab. We figured out how to do these in a very controlled setting, and we figured out how to make quantitative measurements, and we figured out how to really measure what's going on here. And so this is the grand finale. This is the experiment. We took a rectangular, so this is now macroscopic. This is a big experiment. We get rolls of plastic from a supply company online. We cut out a big rectangle, 10 centimeters on the side and 20 centimeters on the long side. And then we just fold this rectangle over and we heat seal 3 of the edges. And the 4th edge is the edge where we're going to look for these wrinkles. And so now we're going to slowly inflate this square balloon, and we're going to watch what happens on the surface of the balloon as we keep raising the pressure inside of it. So there's the crumples. And there's the wrinkles. Okay, so one more time. At relatively low pressure for a small amount of gas inside this balloon, we see these kind of sharper diamond-shaped features lining up along a diagonal right there. And then as we raise the pressure higher and higher, they start to transition into these smooth sinusoidal wrinkles. Okay, and so really the key here was that now we could really be quantitative. So I could take, what we actually did was just go through all of these experiments and look at when you transition from wrinkles into crumples. So we take the sheet on the sphere where the story started, and you can make a measurement of where you go from these wrinkles to these crumples. You can take this indentation experiment where I'm poking this film, and again, you can measure where you go from these wrinkles to these sharper crumples. You can take this cylindrical setup where we're pushing and curving this rectangular film, and you can measure where you go from wrinkles to these sharper crumples. And then finally, on a larger scale, in these inflated balloons, once again, as a function of pressure, we can measure where you go from wrinkles to crumples. And this is when it really became quantitative. I'm not going to expect you to discern this graph. I just want you to see that the data are on the line. This is the big finish. The data is on the line. The data from all 4 experiments follows the same trend. And so this is the clue that we've really isolated the general thing, the thing that works on floating films on water, the thing that works for larger balloons. We found the thing. And the thing is essentially how stretched are you and how curved are you? The y-axis is how curved are you, the x-axis is how stretched are you. And there's a balance between these 2 variables that tells you when you should be wrinkled and when you should be crumpled. And so that was the thing that we got to publish this year in Physical Review X. And we're very excited about that. And to close, I just, of course, want to thank all the people who were involved in this work and my collaborators on other projects, and point out my group webpage and YouTube channel if you want to see some more images or browse some more videos. And with that, I'm finished.
Fascinating.
Thank you so much, Joseph. This was also, yeah, it's fascinating as a research and as images. It's so exciting. Now we'll have 10 minutes break until 7:30 or a little bit more. And then we will start again with the presentation by Rich Pell, and we'll have a discussion. I wanted you to also remind you to pay attention to the chat, because it seems to me that people are really engaged in that, and we'll reflect these questions in the discussion which will follow the presentation. See you in 10 minutes. We are starting with the presentation by Professor Rich Pell, and then we'll have moderated discussion by Professor Edward Morris, and I forgot to actually present him correctly. He is a professor in transmedia, and he is a co-founder and director of Canary Lab, which is a functioning laboratory dedicated to art and ecology within it is in the Department of Transmedia, but it works across departments, and it is one of the places where really artists and scientists meet. So this is our moderator today. And again, I want to welcome Rich Bell, and we're listening.
Great, thank you so much. And thank you for inviting me, and thanks for everybody for showing up and sticking around. Let me get this screen share happening. All right, so I'm in Pittsburgh, Pennsylvania, and I'm actually right now sitting inside a museum that is kind of shuttered at the moment called the Center for Post-Natural History. And I'll be unpacking what that is as I go. But I'm also from Delaware. I am so from Delaware that Jill Biden was my high school English teacher. But this pumpkin is more Delawarean than I am. This pumpkin was bred to be kind of perfectly round and to be almost entirely rind. All right, this is not a pumpkin you want to eat. It was because it was bred for the purpose of withstanding the pressures of being fired out of an air cannon for distances that are approaching a mile. This is an annual event in Delaware called Pumpkin Chunkin'. It's kind of like a pumpkin toss that's gotten way out of hand. But the point is that the forces that have shaped this pumpkin are super culturally specific, right? Not everybody raising pumpkins raises it for this reason. Same species. It's a pumpkin in near Pittsburgh, Pennsylvania, where I am now. And this is in the parking lot of the Sam's Club in Altoona, Pennsylvania. First Saturday every year, except for this year, they have the Giant Pumpkin Growers Association weigh in, and people like drive in these pumpkins from all over the place on their flatbed trucks, and they have like forklifts that carry them around, and they're growing them for weight. You can see this one is 1,182 pounds, all right? A big pumpkin, but you don't get the patch on your jacket until you cross 2,000 pounds. That's where things are at. And mind you, all of these pumpkins were seeds in June, and this is early October. So all of that happens that fast. And the difference between these two pumpkins has everything to do with culture. It has to do with the relationship between nature and culture, the way they push and pull on each other. I'm particularly interested in that little space in between, the kind of the footprint of culture within evolution. All right, and that we can see that in the animal kingdom. Or, you know, these are both adults of the same species. These are dogs. The little one's a Chihuahua, the big one's a Great Dane. And the differences between them, the largest size difference within any species of animal is within dogs, has everything to do with its relationship to us over a large period of time, in some cases, and in some cases over a short period of time. The degree to which we've shaped English bulldogs, for example, is really just in the last century, when they went from having short snouts to having no faces. That happened pretty fast. Sometimes we do it for utilitarian reasons. Sometimes we do it for purely aesthetic reasons. But we do it kind of all over the place. Corn, for example, dramatically different from anything that exists in the wild. And this has been going on for thousands of years. Most of that effort is indigenous populations who are rarely credited. This is Hopi blue corn, for example. Popcorn, all these kinds of like, all that wild diversity goes through a bottleneck in the 20th century, and we kind of reduce it down to like this like very utilitarian kind of feedstock that covers North America that we use for fuels and this sort of thing. What we're doing at the Center for Post-Natural History is we're looking at, we're inviting people to look at a living thing and to see culture in it, and to ask the same kinds of questions you would ask about any other kind of cultural artifact. Questions you might ask about the architecture of a civilization that you don't know anything else about, but you can see how tall their walls are, And maybe you can kind of infer something about what it is they were afraid of. What is it that they wanted? What were their values like? And we're just inviting people to do that with life as well, specifically the life that we have shaped. This is a genetically modified mouse embryo. It's been stained so that the skin is clear and you can just see the bones and cartilage, right? So that's not the genetically modified part of it. The weirdness here is that it's missing a few essential parts. This mouse has no ribs. It's had a single gene that was tweaked, it was knocked out, a gene called a Hox gene. We also have its cousins that have too many ribs, almost going all the way down its spine. That's a really culturally specific thing to do to a mouse, right? That implies a certain kind of reductive thinking. What does this part do? And how can I use that part independent of the whole? So we look at essentially the intentional changes that people are making that are heritable in the living world. So it's got to have kind of ongoing influence through the generations. And this is stuff that we have done on purpose. And this is what sets it apart maybe from larger conversations about the Anthropocene, about just kind of general human impact of the world. We're kind of like a smaller part of that. Like if you take the whole Anthropocene and you just look at the biological components of it, irrespective of climate and geologic influence, and then even within that, you look at just the stuff we meant to do, the intentional parts of it, that's kind of where post-natural history is. Now, what's curious about it is that this is not well represented anywhere. This ought to be a part of the domain of natural history, right? Natural history museums. This is certainly within their wheelhouse. And yet everywhere in the world, these organisms are kind of glazed over, if they're there at all. Sometimes you'll see them, but rarely are they represented in a collection in the same way that you would go out and do field work to try to document things. There's a blind spot there, and it's a blind spot that we've occupied. And I think it's important to ask, like, why is that blind spot there? Why are these things ignored? Lots of reasons. If you ask somebody in a natural history museum, they'll tell you, well, they're really boring. Like nobody wants to see them, which is totally true. And I kind of wonder, is that blind spot, is that sense of boringness, the fact that we don't see a lot of farm animals in our natural history museums, is that maybe kind of intentional? Are we editing ourselves out? Have we been trained to edit out our own influence and our own sense of kind of responsibility in doing that? So that's basically where our name comes from, post-natural history. In looking at natural history museums, we sort of see where they stop paying attention and we go, okay, all right, well then we'll just pick up there. This is everything else, post-natural history. And we've got this museum in Pittsburgh and you can come, it's closed right now, I'm sorry. But give us a few months, let the world figure itself out. We were open a little bit this fall by appointment, and I suspect we will be able to do that again relatively soon. We're open on Sundays, and we have a website where you can sign up to come and visit us. So what I wanted to do today was kind of take two specimens, two birds. We're going to put two birds in conversation with each other, and kind of look at how a natural history museum might see them, and how we might look at them if we were really interested in kind of their relationship to people. If we were to see that culture, if we were to see them as like a lens through which you can look at human culture, what would it say? So this is one that's like not from my collection. So one's not and one is. This is in the Smithsonian in Washington, DC. It's a great frigate bird. They fly all over the Pacific. And this next one is in my collection. This is a yellow canary. This is a Tweety bird, right? And in a natural history context, you know, you've got the label, it's got a species, Fregata minor. We know when it was collected, we know where it was collected. We might measure it and write all that stuff down on the other side of the label. There might be a filing cabinet someplace where you can find out a little bit more about who collected it and maybe who paid for that, kind of the circumstances of that collection. But that's that's about it. Everything else we're kind of inferring based on larger issues of ecology. We're looking at its role in a natural world, irrespective of people. Similarly here, we've got Serenes canaria familiaris. Familiaris usually means it's domesticated. It's a familiar. So we tag that on the end of cats and dogs and a lot of other domesticated animals in their like Latin name. In a natural history museum, this might not even be filed in a place. It might be filed in a special drawer that's just labeled locality unknown. Because it was raised in a cage in somebody's house, therefore it's somehow already kind of tainted in the way that they're looking at the world. It's bad data. So it gets filed away separately, if it even makes it into a natural history collection, which is probably very rare, but it does happen. So this canary, if we stare really hard at it, right, we've seen like it's Tweety bird, right? But if we kind of wander back through its family tree, its ancestry, and we kind of look at like where these first kind of met people, where people started to care about canaries. Takes us to the Canary Islands. Canary Islands, interestingly enough, get their name from the dogs that lived on them. It's canis, same kind of root. The indigenous people there had these like very large dogs, and when they were invaded by Spain, the dogs were noteworthy enough that was the name that the Spanish gave to the islands. But the canaries themselves are kind of like dull green brown. They're kind of dirt colored. They're not these brightly colored animals. All that comes later. That comes from being raised in captivity. So why did we put them in captivity? Why did we capture them? There's lots of animals on the Canary Islands. Why these ones? Because they made sounds that we liked. We really liked their songs. And this is, and I'm using we in we can talk about that later, in a super problematic way. It's a habit, it's a bad habit, sorry. But people that captured them liked their songs, and you could bring them home. It was a way of getting portable music. And people were kind of obsessed with this at this time. This is Athanasius Kircher. This is the first attempt to try to like take bird songs and turn them into like a musical notation. And most of the birds that he's paying attention to on this particular page are domesticated birds, post-natural birds. That's kind of curious. And kind of going down this canary rabbit hole, which is a weird thing to say, I discovered all these kind of interesting twists and turns. This musical instrument's probably familiar to most of us. It's called a recorder, right? If you look up the word recorder in the Oxford English Dictionary, you'll quickly find out that its original use of this flute was for training canaries, training songbirds to learn melodies that people wanted to hear, not ones that their mates wanted to hear. That's what the recorder is. It's for recording music into a bird brain. The bird is a playback device already, right? Pretty soon you get bored of playing the same melody on a recorder over and over again for a bird. So people invented a mechanical instrument called a serenet. That's what this woman is doing. She's staring at her little yellow canary there, turning the crank on this instrument in an attempt to program it, to try to upload human music into a bird brain. This became its own kind of industry, where a whole region of Germany in the Harz Mountain area became world famous once like kind of radio kind of promoted it everywhere for its pre-programmed canaries. You could hear them on the radio, but you could also buy one, and you would buy one that had been already trained. There were rooms filled with many cages and some kind of serenet or a water organ programming human music into these birds, right? And so you have all the kind of stuff that goes into like kind of ideas of copy protection. You can buy the bird, but not the means to train it. That kind of thing were already in play as like an economic function. And this whole story kind of opened up as a result of this one little artifact that appeared in a record store here in Pittsburgh. And my collaborator, Ian Nagoski, picked it up and saw that it was an old record that had been played to death, played thousands of times for an audience of birds, produced as a suitable teacher for roller canaries. So this is like a floppy disk. This is data. And the bird is your storage device and your playback device. To me, this should be the first chapter in like any media history book, but I haven't found one that it's in yet. Canary Records, I got to give them a plug. This is where you get all those kind of recordings of these birds. And I realize I'm talking slower than I should because I'm behind. So the next story, I'm going to try to spit it out quickly. This is that bird back in the Smithsonian, and its story is more complicated and crazier. But if we dig into where this is from, if we look at that label on Johnston Atoll, this is why it caught my eye. It's like, there's no reason for a natural historian to be on Johnston Atoll. That island has been used by the American military for much of the 20th century. It's where we do our kind of craziest things, launching nuclear bombs into space and blowing them up. We actually did this. There's that we again. Let's talk about it. It's tiny. Right off the coast of it is an even tinier island called Sand Island, and that's where this bird was collected from. So what are people doing on Sand Island in the early 1960s? Well, that filing cabinet I mentioned where they have the extra information about like the researchers who collected it, pulled that file. And it turned out that the Smithsonian had been hired by the Navy. The Navy was paying graduate students to go live on Sand Island for a period of time and collect all of these frigate birds. They didn't care about the birds. They were like, you guys can keep them, put them in your collection, put them in a nice museum you got over there, but take all the ticks off of them and send them to us. Send the ticks to Fort Detrick. We're not going to tell you why. So in that folder, I found these photographs where you can see kind of, there's all the birds that they might be collecting. And there's a little sign in the middle that has a date on it. And I want to direct your attention to the horizon where you see these ships, some of the other kind of artifacts of, this kind of military history on Johnston Atoll. But if we zoom in on that sign, it gives like this really great data point, a date and a time. So now we can go back to like the public record and go, what was going on that date and time on Johnston Atoll that these researchers might be collecting birds, and in particular, the ticks that are on them. And thankfully, I'm looking for this information now. Had I been looking for this information 10 years ago, it would not be available. But as it turns out, on that date, highlighted at the top there, you got Phase B. This was a part of Operation Shady Grove. What now we know is one of the largest biological weapons tests that the US ever conducted. And it was over a long period of time where they're releasing, among other things, chemicals and pathogens, including ticks that were carrying tularemia. This is back when we had a biological and a chemical weapons program as a part of the formal military. This was the ship that was listed on that piece of paper. And if we go back to our little picture that some anonymous graduate student took, and we zoom in the background there, lo and behold, there it is, number 40. And this is the only photograph that is declassified of the largest biological weapons test that the US ever conducted. And all that story is in that bird. It's just not, it's not on the label, but it's why that bird is there. So I'm going to end there. I went over a little over time, but that's my story of two birds. And thanks for listening. Let's see, I'm trying to unshare. There we go. Have I unshared successfully?
Yes. Yeah.
Oh, good.
Thank you so much, Rich.
Yeah, wow. Each of those presentations was so engaging. So my job is always, there's lots of great questions in the chat, and I'm sure all of you have questions and I have specific questions, but my primary function here is to kind of find a commonality or to put these various presentations into some kind of conversation with each other before we turn to these specific questions about certain people's presentations. And each one was so captivating, it was hard because I was so focused on each one. But here's the axis that occurred to me as I was listening to each one, is that that each presentation to me had an interesting relationship between the question of utility or some kind of function, and aesthetics and some kind of pleasure. That is, that each person in their presentations, whether explicitly or implicitly in the way they presented, expressed these values, the value of some kind of function or utility and some kind of expression of just pure pleasure or maybe aesthetics in the work. With that, let me try and kick off the questioning. Oh, the other commonality I found was that each one is kind of a detective in some ways. So you're trying to find out something. And there's sort of detective work going on and some kind of aha. I'm particularly interested in that because I used to be a private investigator, which is another topic of discussion. This is a true story. So let me start with asking each of you in a slightly different way, something about that question of function and pleasure. And in particular, I want to start with Jennifer and Rich in terms of asking you each kind of why you think you've chosen, if you have chosen, I suppose, to frame your work or position your work within the cloak of an institution, a museum in one case and a lab in the other case. Both the museum and the lab would seem to imply some particular type of function or service. You know, what that does for the work or what your motivations were for doing it. Maybe just start with Jennifer.
Sure, that's a great question. So if I think about the decision to build a lab, there's a few reasons for that. First of all, I was really tired of traveling. When you're an artist in bioart field, you end up traveling a lot because you're begging scientists to let you in their lab. And so it's like you travel to them, and then you also try not to wreck what the primary focus of that lab is, which is their research. And that's a little hard sometimes because you want to like dance on the countertops, or you don't know how to not contaminate the incubator yet. And so there was like a real functional rationale for that. I wanted the primary function of this space to be art, and then other things could happen on the side. I didn't want to travel so much. I didn't always know what country I was in when I woke up, and I found that a little stressful. Then it also became it became over the years a form of institutional criticism, but then maybe even institutional proposition. So now it's become this like desire to push the limits of what a lab can be and how it can function. And so I would say some of my greatest performances in my life have been for health and safety officers, or people who can provide certification or permits or these types of things. So, and at the beginning, that was like a side effect. And now I think it may become the essential thing. And one of the things I'm collaborating with this other woman from the Healthy Headwaters Lab, so she's a scientist, but she's also looking at ways that science is entangled, it's cultural, it has a politic to it. And we need to reassess those strategies. So she's creating new methods in her lab for how to do ecological sciences with that understanding. And I think I'm doing the same. I think our labs are both exploring how to engage in scientific protocols, procedures, practices, but then also address the other components of the universe that are not measurable and have so much other things to offer. So I actually think sometimes it started off as a function thing. It became kind of beautiful and fun. I like also it was a fundraising strategy because it's harder to get money as an individual than it is if you're an institution. And then the last thing I will say is that when I was young, I felt like this sort of crazy hippie, and people wouldn't always take me very seriously, or they were very fearful of what I might be doing in a lab. And now that I run an institute, it gives me a lot more authority to sort of propose these very absurd things.
Laura Heyman had a follow-up question on that, which she was posing the question of whether, in this case, a lab is analogous to an artist studio.
Totally, and especially the second one is really an artist studio lab combo. So now, like you get into it and you're like, wow, this is kind of cool. Maybe I'm exploring new architectural spaces and what those could be like. So it's sort of, it's really evolved. And I would definitely say, even though our lab is a certified lab, it's a theater, it's a performance space, it's an artist studio, it's a kitchen, it's all of those things.
Yeah, I want to come back. I want to circle back at some point to this idea that you mentioned in the midway of your answer of kind of how the lab maybe provides a proposition, particularly with respect to maybe the university at large. But I want to get other voices in here too and get,
sure, go ahead.
Turn it to Rich. So I guess the same question to you in terms of why you sort of found yourself couching your practice in terms of a museum, and what you feel like what avenues that seemed to open for you, or kind of why you ended up there.
I mean, it's a little paradoxical, is I'm wanting people to be skeptical and critical, and yet I'm kind of using a lot of the trappings of the authoritative voice in doing so. And I find it functions really well when I'm present in the museum, because I don't think I project a whole lot of authority. Like, we can have a conversation, we can kind of get into it, and it gets complicated quickly in a way that it doesn't when it's just kind of there presenting itself. So I take ownership of that. But what it gets me is to be a peer with other institutions. Other museums see me as an equal, or at least they can like play along long enough to get me in, and then I get to be in their collections, and they get to be in mine. Like, my stuff can go over there now. And so, before I opened the museum, that was like kind of an impossibility. I couldn't, I don't have any degree in anything that would be meaningful to a natural history museum in terms of science. But now it's pretty easy. I can just ask, and and I can go and I can look in their collection. I can propose, hey, I'll bring some of mine and you'll bring some of yours, and we'll do something together. And I'm afforded a level of access that I couldn't have imagined before, just by kind of performing an institution. And at some point I've become one, but it's not clear where that happened.
Yeah, I wonder about that. That's a Rubicon that you'd like to recross when you became an institution rather than performing one. That's related to the follow-up I wanted to ask about Jennifer. So let's hold that in reserve. And I just want to turn to Joseph here to get you in here. So it's interesting, because to me, listening to this, because in some ways, Joseph, your presentation was this is clumsy to say so, but it's sort of the inverse of what you might expect in terms of the ratio between expressions of pleasure or pure curiosity and expressions of function. And there were lots of questions in the chat about why, what are you doing this for? What's the application? And so I do wonder about the application of these experiments and discoveries that you've made, particularly maybe for engineering and so on. So that's the second part of my question. The first part of my question is that true? Is that how you go about your work? That you're motivated more by sort of a love of knowledge and uncovering things, or are you motivated by finding real world applications to the things that you're discovering?
Mm-hmm, mm-hmm. Yeah, so great questions. So I would say that as a graduate student, actually even as an undergraduate, when I was choosing graduate schools, in my own sort of personal path, I gravitated towards science where aesthetics were a strong component of the science. And so soft condensed matter is a subfield of condensed matter, which is a subfield of physics. And inside of my little niche subfield of soft condensed matter, there's a lot of sort of folks like myself who are really doing curiosity-driven stuff. That's I think where aesthetics plays a big role. So yeah, that's my flavor, and I knew this was an audience where I could really have fun with jazzing on the kind of stuff that gets me excited about the work. In some ways, I really liked your opening question, because I would say that in some ways I try to cloak my work inside of a lab. It's fun to push boundaries of what falls into the umbrella of science. And so sometimes sometimes you present your work in a certain way, but you try to push it a little bit further and try to get different questions or different ideas into the subject.
Have you ever, oh, go ahead, finish the answer.
Go ahead. As far as I can tell you about applications, but
yeah, go ahead. Yeah, answer about the application. I think people are dying to know, like, what are the engineering applications and what are some of the applications of this wrinkle to crumple discovery?
Yeah. So it is curiosity-driven science. I am funded by the National Science Foundation, and they have a style in their funding agency where they encourage fundamental science that doesn't necessarily have a particular application in mind. With the idea that curiosity-driven research often leads to breakthroughs that will in turn become applications. But to give you a flavor of what some of those possible applications eventually may be, some of them are like smart surfaces. So you can make a surface that maybe you want to wrinkle it or unwrinkle it, and it will become water loving or water repelling. Or sort of texturing of surfaces that you can actuate on and off with temperature or with light. So sort of smart materials is one kind of category. Some of my talk touched on inflatable structures. So I was talking about these wrinkles in balloons. NASA cares about inflatable structures because they make really good payloads for shooting up into space and then inflating it once it's in orbit. So there was an experimental satellite that was actually an inflatable balloon satellite that had been developed about a decade ago, as well as high altitude research balloons. So sort of ballooning is one aspect. And there you want your crumples to be where you want them and to not be where you don't want them. So it's sort of a structural engineering kind of perspective of where your thing is going to crimp and where it's not going to crimp. How do you design your balloon in a way where it functions how you want it to?
It's also such a delight in listening to you talk that we're talking about crumples and wrinkles as opposed to some words I've never heard of before and have to quickly Google to figure out what they mean. But it's refreshing to understand that there is a significant funding source for scientific research that's, as you say, curiosity driven, and your articulation of what could come out of that, and that is a good segue, I think, to circling back to this statement that Jennifer made about the lab becoming a proposition at some point, and also tying in, Laura just put a comment in the chat about why, if aesthetics is a frame of the research, or if curiosity is a frame of the research, why there's not, more collaboration between art and science, like true collaboration. I do find that oftentimes when art is brought into a science context, that what science wants out of art is to be like the jazz hands of science, or somehow to aestheticize it and jazz it up in some way, and that's usually not what artists are interested in. But all three presentations do indicate some ideas of more substantial collaborations, or maybe even a larger proposition about what the academy is or can be with respect to this interface between the sciences, the humanities, aesthetics, and so on. Is that the neighborhood of considerations that you're thinking of, Jennifer, when you talk about the lab as a proposition, or what did you mean by that, the lab as a proposition ultimately?
So coming up through the ranks of like critical theory, a lot of our ways of engaging with science was critique, right? And I started thinking that it just seemed like a real Debbie Downer all the time, and that maybe one of the things that I could do is propose alternatives, right? So I'm really thinking of this as like a hyper-local, feminist, intersectional, community engaged lab space, right? Like, what about just doing all the opposite of the things that we read about in philosophy of science that we're so often critical of the hard sciences for, and to just try it. And so that's what I mean by propositional. Rather than offering a critique, I'm proposing alternatives. And it doesn't always work, and it's like a total messy work in progress. But I want to try new things. I want to imagine new futures. So another example that would be with the biotech futures. So many scientists I know, and also like people in legislation, like lawmakers, rely on the metaphors from like literature and films to help them determine biotech futures and what the law is going to be and what we need to be concerned about. So what if we start making these other stories, these other science fiction stories that propose futures that we would like, and futures that are egalitarian, and futures that acknowledge all of the knowledge that's come before us and all of the things that are unmeasurable. So I think that's what I mean. I'm trying to offer up alternatives and suggestions and actions and like, you know, really taking action rather than being critical of the action I see others taking.
That distinction between critique and proposition making as it relates to art making is extremely interesting to me. I think there is a kind of modernist or postmodernist hangover where critique is considered the primary function of art. And I'm happy to hear you talk about proposition making in that way. You know, Rich, you teach in an art department, is that right?
Yep.
Do you see things in a similar way, or how related is the Museum of Post-Natural History to the university? Is it related, or is it not brought within the university?
I've kept them separate so that one is not beholden to the other. But I do teach, and I teach, you know, post-natural history within my classes in a lot of different ways. Sometimes it's like a museum's intervention course, and this is just kind of one example. But sometimes we delve more into like the science of things. But yeah, I totally agree that as artists, you know, we need to be getting our hands dirty and risking being kind of entangled rather than just always being kind of in the safe space of critique, where you're in the safe zone if you're just always on the outside pointing. But I think the really interesting things happen when you're risking affecting change and being kind of implicated in whatever comes next.
Yeah, and do you see a relation? So to me, the macro trend within the university is the increasing marginalization of the humanities because they can't be instrumentalized or monetized, which relates to the conversation I was just having with Joseph. And so I don't know if there's really anything you can do about that. Sometimes it feels like fighting against the jet stream or something, but like, I wonder if these models, if you feel like there's any hope in these models that are being presented here tonight in terms of providing a more sort of productive and integrated vision of the humanities and the arts within the university.
I mean, I guess I tipped my, oh, sorry.
No, you go ahead, you go first.
Oh, yeah, go either one, yeah.
Okay. Yeah, just briefly, I guess I tipped my hand when I said I keep these two things separate, because I don't trust those models when it comes to like making something work over long periods of time. I find institutions to be very fickle in terms of like the rise and fall of funding, and especially when it comes to art and something scary happens, universities will disappear real quick and try to disappear your project with it. And I've just seen that happen to too many others. So for my purposes, I'd rather keep things lean and outside the institution, but that's a privilege of being employed also by the same institution. It's not a shell game that everybody is able to play.
Did you want to add something to that, Jennifer?
I totally agree. And I've kept some parts of my life different, like are segmented in different ways. There was a period of a time where a year I wasn't employed by a university and I ran around naked everywhere I could because then those rules didn't apply, right?
I've seen the pictures, they're online.
Oh, they're on my website. I'm worried about, it's a process called reification, right? Where things that are outside of the norm get pulled back in and maybe reified or transformed or used by center or the power structures that be to continue to meet the goals that they intend to meet, right? So it could be like capitalism or a large institution or something like that. So I do find that whenever I think I'm staking a claim on the periphery or outside of the established norm, so often those things get sort of co-opted and used maybe in ways that I hadn't intended. And I don't mean me as an individual, I mean us as like a creative generation or whatever. So for example, I remember when I was very young, one of my first projects was a fictitious corporation. And we created all sorts of media campaigns and a website and printed banners, and everyone thought we were a real company. And but then now, like 20 or 30 years later, that same process is being used by big companies because they're like, it's cheaper. We can make disposable buildings that all have this plastic crap on it that makes it look like a system of authority. So I feel like there's this double-edged sword. Sometimes right now, I do think that bioart is doing very well. It helps integrate the humanities into these other genres. But I also worry about what are the unintended consequences. I have no idea what they are, but they're coming.
Yeah. Joseph, I want to get you in this conversation too, because I'm wondering, you know, you're the type of, it strikes me that you're the type of a person who would really enjoy these types of collaborations, these types of spaces. Have you ever collaborated with an artist before? Is that something that interests you? And how do you think that might look like?
Yeah, I haven't really done it. Other than the fact that my wife is an artist, she does landscape art. So, you know, I get a little window into her work. But as far as getting my lab involved in art, I guess when I was a graduate student, there was a pretty active art and science collaboration at the University of Chicago. So my PhD advisor was working with an architect, Jeannie Gang, and they were doing some fantastic stuff together. So, you know, I've seen it in my community. It's going on in my community. I would say that is something that I'd like to do. It's always a function of limited time, but yeah, it's something that I'm definitely interested in the future.
So I don't want to. I pursued, you know, this line of thought here. I don't want to dominate the conversation over much. That's in some ways my job. If it's silent, I do dominate. But I do. Let's make sure we get to some of the questions in the chat before we move on to the nuances of this discussion. Does anyone, before I go to back to the chat, does anyone have any questions they're burning to ask on this point of view? Yeah, Laura.
Yeah, I have a question about this, like, you know, part of what we're talking about, what nobody's saying is money, and that the sciences receive funding and the arts do not. And how can the arts receive the same kind of funding that the sciences receive? One of the ways seems to be to align yourself with the sciences. Another way, if I'm thinking of another kind of Rich, if you were saying for artists to be engaged in like making change or like whatever the results of, rather than standing in the back and critiquing, but getting involved. Like, so artists aligning themselves with, for lack of a better term, social work, right? Social practice, which has exploded in the past like 5 or 6 years, 7, 8 years. And the majority of funding in the United States is directed towards that type of practice. But the arts in and of themselves, for that kind of, you know, curiosity-based research that Joseph is talking about, which seems to be like central to the aspect of being an artist and also central to the aspect of being a scientist, is less possible, right? Like somehow that's not, so it's not that I don't know the answers to those questions. I'm like so many of the parallels here and the problems and this kind of skirting that you started this whole, that you started in the beginning of the chat, Ed, is like something that artists are so, like, that's just a constant thing, right? For artists, that skirting, that bobbing and weaving, whatever. That's not really a question. Sorry, I'm sort of spitballing.
No, it's a great comment, actually. It deepens the conversation we were just having and does make it real. I mean, I did mention the fact that, you know, arts are being marginalized in the university because they can't monetize their work in some way. And so that's why it's interesting to me that there's an avenue for funding, presumably even greater funding than arts have the capacity to get, for curiosity-driven science. Probably because the assumption is at some point down the line that can be monetized or instrumentalized in some way. So even though, you know, Joseph is proceeding from the motivation of curiosity, at some point someone probably has the idea that this stuff can be instrumentalized. I guess that to me, you know, my point of view on this, and I'd like to hear from anybody, a panelist or anybody, that this is sort of a, you know, I think that it's not a productive train of thought to blame that on science, and I don't think you're doing that. But it does strike me that art could learn something here in terms of forwarding or foregrounding or giving or embodying some of the principles that I think Jennifer was talking about in terms of making propositions as opposed to critique. That some aspect of the marginalization of art, and not fully, but some aspect of the marginalization of art has to do with its, in my view, this is totally arguable and controversial, it has to do with its doubling down on critique as its main function.
I think the main issue with art is its inability to be instrumentalized or monetized in a mass form, right? Like capital is the problem, right? Every artist can't follow this model. I would say that's and not to discount the issue of critique, but again, I would point to social practice, which is a like largely utopian framework where people are trying to imagine as broadly as possible, similar, Jennifer, to what you're talking about, to imagine a future as broadly as possible with as few strictures as possible in the best light possible. Really, like that's what that movement is ostensibly all about. Whether it actually fulfills that stated claim is an enormous question. But yeah, I mean, I don't blame this. I wouldn't say I would blame the sciences for that. It's not, but the inability of art to be monetized, the inability of the humanities to be monetized, the inability of like someone writing a novel to be monetized at the scale of the person who invented styrofoam. You know, it's just, there's no parallel. We couldn't even begin to imagine a parallel.
Do any of our panelists want to respond to that?
I mean, I wish I had an answer. You know, I think as a younger person, I had a pretty knee-jerk, like, reject the funding, you know, to work outside of all the institutions and stuff. But that was like coming from like a total position of privilege where like I obviously didn't need it, you know. But I you know, not to say there's a lot of money happening. I just kept things within the means, and you know, I've worked that way, and now I have a job.
Although both you and Jennifer did talk about the ability to get funding by framing your art within an institutional framework, and I can also attest to that as a strategy.
Yeah, it used to work. It hasn't worked for me in years.
Yeah. So I don't know, maybe we should switch gears a little bit here and see if we can circle back to this. Tom Sherman has a question about, he'd like to ask Joseph about microscopes. Tom, do you want to ask that question?
I typed in the chat, I typed it in incorrectly. I was really interested in when Joseph was presenting, he talked about macroscopes. Mm-hmm. He talked about using SLR technology, et cetera, et cetera. I think one of the things that we're touching on here is that oftentimes there is this almost a privilege of science to deal with microscopes, right? Actually, it's kind of funny because Heidi, she had the best microscopes when she moved to SU. She brought them from SUNY Upstate, and I thought it was kind of interesting. And usually science is kind of based in these, you know, powerful tools. So the idea of using in your research SLR technology to kind of get the data that you're doing, you're grabbing images, you're doing analysis, et cetera. It seems kind of different. So I wondered why you decided to go that route rather than, you know, material specialists are dealing with microscopic information all the time and probably wrinkles and crumples and things like that. I mean, but microscopically, I just wondered what your strategy was.
Yeah, thanks for the question. So I think for me, this comes back to this theme of aesthetics that keeps coming up, which is I was personally drawn towards research that was more ideas driven and less techniques or instrumentation driven. So, you know, there's tons of science out there. A large chunk of the pie chart, where people are making progress only if they have the state-of-the-art tools, the state-of-the-art instrumentation, time to access facilities that have, various this, that. I think, again, this is something somewhat unique about my niche inside of my research area of soft condensed matter, is that the work is very, very ideas focused, so that the challenge is not so much to make a hard measurement with an expensive instrument. The challenge is to ask the right question that can be usually answered with a cheap camera and a dish of water and a piece of plastic. So yeah, and it's what I was drawn to. It's a small segment of the scientific enterprise, but it's what I was drawn to.
I thought it was also interesting that, Rich, when he's making his presentation, was dealing with these species, not from a, there are divisions in biology departments, and everything has to be genetically analyzed. There's hardly anyone that's dealing with the morphology of animals anymore. At least certainly at Syracuse University, it's kind of a lesser thing. So I was very interested that you, obviously by doing your talk culturally, you were dealing with the appearance of these animals, Tweety bird and all that kind of thing. So I thought that was also an interesting thing, because I'm a big proponent on macro view. I think it's seeing the way we see is really important. I've tried to do some work with bacteria. And, everybody keeps telling me I have to get these microscopes, et cetera. But bacteria make, great displays that are macroscopically and easy to analyze.
Boriana, you had a question.
Yeah, so my question, I'm always interested in the way how people create or how they research subjects of their interest. And that's why when I watch biopics about artists, I'm always very disappointed that most of the scripts circle around their love stories as opposed to the way how they develop their art. It's just like, where's their lover? And now when I was listening to Joseph and to his presentation and these big containers, wine containers, I was just wondering how exactly, it's a question for everyone, but first for Joseph, how do you find your wrinkles? How do you find these things that become a subject of your study? Is there a chance in it? Is there also, like for me as an artist, it's very important that I spend time doing nothing. If I do nothing, this is the most creative time. The most interesting ideas come to me in that way. So it's about protocols of study and protocols of finding things that are interesting.
Yeah, I also wonder, Joseph, if you've just become like totally paranoid about seeing patterns everywhere.
Yeah, I mean, it's definitely a creative process where these crumples, came about from a lot of different directions. And I guess the seeds were planted pretty early on, attending some seminars, talking to some visitors to some labs. You're definitely right that you have to budget some time to do nothing and to not plan on getting a productive element out of some activity. Because yeah, the Crumples project was definitely not planned. It was sort of a matter of part serendipity and part just following up every time some clue came along. So yeah, it's not the typical project, but it's one that was very, very enjoyable. I mean, the wine tanks came from a research article that I stumbled upon after working on this for many years. So, that stuff I really liked in Rich's talk, finding this ship in the background of this photo with the date near all these birds. I mean, that's exactly where this wine tank just appeared in a review article that I was looking through from a website that was cataloging all the shell buckling images that this person runs a website of everything that has ever buckled, everything that has ever failed. There's a website called Shell Buckling, and I was just browsing shellbuckling.com. I found these wine tanks and I was like, oh my God, those are crumples again. So yeah, you gotta browse Reddit. You gotta just do stuff that's not productive in science and everything.
That's a great question. I'd love to hear everyone talk about that a little bit. You know, in terms of I heard in the chat before everyone joined, we were talking about COVID and what the sort of downtime has meant to people's creativity and so on. So yeah, is the gist of your question, Boreana, am I right to interpret as the value of doing nothing for periods of time, or is that right? Or yeah, okay. Rich, what is the value of doing nothing for you?
That's my meditative practice. I mean, to me, it's all about like looking to the periphery, like looking at the things that other people are filtering out, you know? So for me, that's like the boring stuff. If somebody says that's boring, I stare really hard at it. It's a provocation because it's hiding something. Boredom means like there's nothing to see here. And I think that's true about the wrinkles. You know, you're not supposed to be staring at the wrinkles. And that's where the good stuff is like peeling the blinders back further than you're supposed to, you know?
Is that like a max? Are you ready to say that's like sort of a maximum or a principle? Wherever there's boring, there's maximum interest. Is that going too far?
I'm building up to that. You know, but yeah, I'm suspicious of that. If something is boring, I'm suspicious of why.
I have a couple principles that I use to sort of generate ideas. First of all, I have a really big imagination, but it's kind of like a 5-year-old. So in my mind, I'm like, and then, and there's always like, and then there's a Mountie, and then he's dancing. And so I have like these like really elaborate stories in my mind that often generate the events that I host or the projects that I make. I tend to write them down because I have a very bad memory. And also they take many years to execute because again, I've become this like institutionalized artist who does these really like makes institutions do things they don't want to do. So it takes like many years and some of it's money, but some of it is also like the paperwork and convincing the fire safety officer that this meets their criteria or that type of thing. The other thing I have to say is that I spend a lot of time online shopping and tinkering with medium to older technological objects. And I just like, I'll play with something and figure out what it's supposed to do, but then maybe what I want it to do. Like I would say, in fact, in a lot of my grants, I say that I am producing new and novel uses for existing technologies. So that means that I'm buying like shower curtains and like you should see my Amazon list. They don't know what to recommend to me because I buy the most absurd range of stuff on Amazon. And then I'm at home and I'm like figuring it out. And then it occurs to me that that's why. So right now I'm collecting snow globes because snow globes really contain microbes really well and they're safe to take outside of the lab. So I have every snow globe in town on order coming to me before they run out at the end of Christmas.
That's great. So I'm tempted to go to there's a number of questions that are very specific to people's presentations, and I try and hold those to the end, but I don't want to disturb the flow of any conversations. Anyone else have any questions for all of the panelists before I go to some of these questions in the chat? No? Okay. Joseph, there's a lot of questions about kind of what these, sort of what conclusions or what these wrinkles indicate. So just to take one that's in front of me here from Debdrita, she says, I was just curious, have you watched these wrinkles on water surface? Have you tried these on any other liquid medium? Are these patterns different or do you think they would be different on other liquid mediums?
Mm-hmm. Mm-hmm. Yeah, so let me try to answer that in a couple of ways. So there's many aspects to these patterns. And to give sort of a condensed single kind of talk, I focused a lot on wavelength. And so the wavelength, or the width of one wrinkle, that will usually tell you a little bit about the sheet and a little bit about what it's resting on. So water has a certain gravity, or a certain density, which tells how gravity affects it. So if I had put a little bit of salt in the water to make it a bit more dense, then the wavelength would have shrunk a little bit. If I put it on a less dense liquid like alcohol, the wavelength would have been a bit bigger. So So yes, the liquid definitely has an influence. And what I hid in the talk were all of the equations that quantify all of that. But absolutely, the wrinkles are a result of the sheet doing some compromise with the substrate. Yeah, where the water is the substrate for all the things that you saw today.
Does that answer your question? So Devadvita also had a question for you, Jennifer. She was really drawn to the mitosis, if that's the right word,
yes.
in your artwork, and just want to know about the technique, right?
Yes, that's right.
Go ahead, Devadvita. Yeah.
I work in a cell biology lab, and I am thrilled about mitosis because that was one of the things that I study. And that was one of the slides you went through like really fast, like swoop, swoop. And I was like, wait a minute, just hold on right there. So yeah, I really wanted to see it. Yes, exactly, that one. So what techniques do you use here, and how do you make these? This is incredible. This is beautiful.
So these are Petri dishes that have agar in them, and we've developed a variety of techniques for staining the agar or making 3D agar forms. And then we do also, some of them are temporary tattoos. So that bug on there is a temporary tattoo on the surface. There's some sequins, there's some 3D casting. And then we put collage materials. These cells, this is a mitosis diagram that I've cut up and stuck right into the agar. And then once they're grown to the point that and then I seed them with GMO bacteria. So these are all GMO bacteria in here. And then I put them on top of these paper collage materials that are these theater sets. And then we do top down like StatCam photography of them in the lab. Does that answer your question?
Yes. So I had a follow-up with this question is how long do these stay? I mean, because, after a couple of days, the agar kind of starts to shrink.
So we have found windows, and because you got to time it right for the day you've booked a photographer, right? And the day you have time to do all that. So I find that I also go in and check them, and sometimes I heat them up a little or cool them down. But these are usually, I would say, 5 days, maybe 7 days of growth. Yeah, and then they die, they stink, they smell terrible. I've experimented in the past with dehydrating them and then fixing them, but I found a new recipe for just fixing them without dehydrating them, because when you dehydrate them, they're not as, you lose a lot of your detail. So eventually I would like to fix them in resin if it's possible. I will stop sharing my screen though now, because I don't want to dominate the visual field.
Yeah, it's beautiful. I just got so drawn to it. It's beautiful.
Thank you. There's 20 of them. I'll put my website on the chat if you want to check them out.
Yes, please. Sure. Thank you.
Thank you.
So just turning to another question in the chat. It's from Amra. Is Amra still around? Do you want to ask the question, or do you want me to ask it for you? You want to go ahead? You're muted.
I was just going to ask which one for which speaker.
Oh, sorry. Yeah. Oh, sorry. Right. So I'm looking at one that asks, I think, Rich about his thoughts on genetically modifying organisms.
Oh yeah. So basically, it's more of a question of intention. Like, does it matter whether all these organisms are being genetically modified for like gaining knowledge or for capital gain? Like, what are your thoughts on that?
Are you asking me as like an ethical question?
Yeah.
Okay. Yes. Short answer, yes, totally matters. I think everything needs to be like weighed on a case-by-case basis. Mm-hmm. I mean, that was kind of the thing I learned more than anything in doing this project. I think like way back, I like started wandering into this realm like almost 15 years ago, and had like a pretty clear idea of what was good and bad at the time. And over time, it's gotten much more complicated. And I feel like my role has become more of like, how can I get people to think longer and harder about these things more than anything else? More than like their ultimate choice that is made. How can we learn to give our attention over to problems that they don't resolve themselves easily into like a good or bad. So yeah, the short version, I guess, is I don't have a short answer to that question.
Well, it's interesting to me, Rich. I mean, in some ways, I feel like the work through the Museum of Post-Natural Histories is a museum or center, actually?
We're a center, but...
Center, right. Okay, but it reminds me of the Center for Land Use Interpretation in a way. I don't know, that's probably an analogy that's been made before for you, but, and there does seem like a conscious effort to remain unrhetorical, say. Is that accurate?
Yeah, we wouldn't exist without them. I learned it by watching them, 100%. They are, they maintain sort of a facade of neutrality. Like they're not neutral as in like, like no museum is neutral. And like I adopted that to a degree, but I found I couldn't really maintain the facade so well. But I think what I have taken from that is the pedagogical value of me not telling you how I think you ought to think. Like that's a part of my job, is to lay out these really complicated problems that are like puzzles. They're like missing a few pieces, and to withhold how I think you should walk away from it. That's my way of getting people to think harder.
Well said. That's a difficult balance to strike sometimes, I find. Yeah.
Super.
Well, look, this whole event is now gone past 2 hours. I'm happy to stay here, but I also want to let people know that you're welcome to start disappearing. But there are some unanswered questions in the chat, but they're mostly specific questions. I think that those types of questions maybe could be emailed or answered to people if they have them. Do other people have other questions that they're really dying to ask at this point that feel like they've been unanswered? I see no one rushing into the gap. So what do you think, Boriana? Should we think about wrapping this up or what?
Yeah, I think if there's no more questions, we can wrap it up. I think it was, all the talks were amazing, and I think we had a very interesting discussion, which I think will spark probably more questions after we finish. At least I'm thinking that I'll be thinking about the things presented here a long time, along after it. Yeah, there's some people started to, I think there is one question here.
Yeah. Oh yeah, someone wanted to give a link to the canary bird sounds. Someone else had a question early on that actually was fascinating to me too, Rich. It was like, is there anything that existed for matching the tones, like an instrument to match the tones of the canary, or was it just the melody?
Oh well, yeah. I mean, there are bird calls, right? There's a whole class of instruments that's about bird imitation. Mm-hmm. And then there's the whole art form of using, just the human body to imitate birds. And so I'm pasting into the chat window a link to Canary Records. They have several collections that are dedicated to bird imitation, exquisite bird recordings, as well as the bulk of what they do, which is the music of immigrants to the United States, 19 teens, 20s, and 30s. So non-English language music, virtuosic performances in New York City by Turkish-speaking people long before the first country record was recorded, right? This is music happening most often in America, but has been erased from our history of America. So that's their primary job. But then they also delve into birds and our relationship to birds in really beautiful ways. They have an entire record dedicated to duck calls. I love them so much. Their proprietor, Ian Nagoski, I met him at the lunch table my freshman year in high school, and I wouldn't be thinking about any of this stuff if it wasn't for him.
Good to acknowledge our guests. So yeah, thank you everybody for your amazing presentations, and for Boriana and Heidi for putting this together. As always, this was fantastic. So thanks everybody for joining, and yeah.
Thank you. Thank you, everybody.
Sure.
Thanks,
Thank
everyone.
you so much. Thanks for having us, Boriana. It was wonderful.
Thank you. Thank you for joining. And again, for those who came a little bit later, we record our meetings, and they're up on the YouTube channel of the BioArt Mixer, which can be found also on Facebook and in all of the advertising we did before our meeting today. Thank you again.
Cheers.
Take care, everyone.
Boriana.